Dear students, in this video we will discuss about DNA sequencing. What is DNA sequencing? DNA sequencing is the process to determining nitrogen containing bases in DNA. What are the significance? It is important to know the sequence of gene, its impression, protein interaction to a particular DNA segment, same for regulation. What are the methods by which we can do it? The first one is Sanger method. It is also called chain termination method. Second one is Maxim Gilbert sequencing. And third one is automation. Before discussing on the Sanger method, we should know about Frederick Sanger. Frederick Sanger won the Nobel Prize in Chemistry twice. The first time in 1958 and again in 1980. The 1958 award was given for his work on the structure of the insulin molecule and the 1980 award for determining the base sequence of nucleic acid. Now, Sanger sequencing was developed by Frederick Sanger and his colleague in 1977. We know very well for the elongation of nucleotide chain, deoxynucleotide, that is ddNTPs are required. But in Sanger method, a most critical reagent that led to advancement in this method was dideoxynucleotide, ddNTP, because there is no oxygen at 3 prime, so further polymerization will not take place and the chain will be terminated. Now let's discuss the procedure. First, single stranded DNA will be sequenced, for which tuck DNA polymerase will be used. Second, primers will be used with DNA polymerase. Third, we'll use 4 different types of ddNTPs, which are ddATPs, ddTTPs, ddGTPs, ddCTPs. Fourth, in one tube only one type of ddNTPs must be used. Hence, termination occurs at a particular base. Fourth, ratio of dNTPs and ddNTPs maintained in such a way that all type of possible fragments will be obtained. So, for Sanger sequencing, we should have single strand DNA, which we can get by heat treatment. Suppose DNA segment is AT, GC, GT, AT, AC, GG, C, and the polymerase will move from 3 prime to 5 prime. This single strand DNA will be taken in 4 different tubes. In these tubes, along with primer and DNA polymerase, we will take in dNTPs, DATP, DGTP, DCTP, DTTP, and in each tube we should have only one ddNTPs, such as in first tube we are having ddATP, second ddGTP, third ddCTP, and fourth ddTTP. After the process, we can get the different fragments. In first test tube, we can get TA or TACGCA, TACGCA, TA. It depends when ddNTP is attached on growing fragments. Second one, TACG, TACGCA, TATG, TACG, CA, TA, TG, CCG. Third one, TAC, TACGC, TAC, GC, AT, AT, GC. And last one, only T are TA, CG, CAT, TA, CG, CA, TA, T. Now, after getting the fragments, we have to run these fragments in polyacryl amide gel electrophoresis and will get bands. Lowest position single nucleotide band T, then second position TA, third position TAC, and fourth one TACG, and so on. It means we will get the sequence easily by observing band sequence. This sequence will be complementary sequence of DNA, and by this way we can know DNA sequence. Let's discuss a question. Which one of the following set of essential components are required for Sanger method of DNA sequencing in a required buffer containing magnesium chloride and tris HCl? First, DNA template, a primer, 4 deoxyribonucleotide, 4 labeled dideoxyribonucleotides, and DNA polymerase. Second, DNA template, a primer, 4 labeled dideoxyribonucleotides, DNA polymerase, and DNA ligase. Third, DNA template, 4 deoxyribonucleotides, 4 labeled dideoxyribonucleotides, DNA polymerase, and DNA ligase. Fourth, DNA template, a primer, 4 labeled dideoxyribonucleotides, DNA polymerase, and telomerase. The correct answer is first one, labeled dideoxyribonucleotide by P32. Now, thanks for watching. For more information, subscribe, like and share this channel.